Hey guys, my name is Cooper Wade,
Michael Bates.
and we're going to review biochemistry, essentially the building blocks of life today, to help with the AP Biology or whatever biology course you're currently studying. So the first thing we're going to talk about is the properties of water. Real basic 5, you have its attraction to other polar molecules. It stores heat, it has a high specific heat, it has a high heat of vaporization, a low density of ice, and high polarity. So the structure of water is 2 H atoms joined to 1 O atom by a covalent bond, which means that they share electrons. And water's polar. So that means that it has opposite ends with opposite charges. It's polar because the oxygen here pulls electrons much more strongly than the hydrogen atoms, thusly making it polar. Water molecules are attracted to each other by a weak hydrogen bond, as well as being attracted to other polar molecules, which allows them their solubility. And a solvent is what does the dissolving, and a solute is what is being dissolved. Water is actually known as a universal solvent because it's capable of dissolving more solutes than any other liquids. In the terms of pH, water is considered basic, which is a 7 on the pH scale. As the pH scale goes down, it gets more acidic, and as the pH scale goes up, it gets more basic. Water is neutral, so this would be a strong acid, this is water, and then a strong base would be somewhere up here around 13. So, basically you have the 5 main elements that make up most of life. You have carbon, hydrogen, oxygen, nitrogen, and phosphorus, known as the chont in biology. And so they build up macromolecules, they build up the functional groups, and we're going to go into that a little bit right now. So the functional groups, these are the groups that play an important role in the formation of molecules like DNA, proteins, carbohydrates, and lipids. Essentially what your functional groups are, you have hydroxyl, methyl, carbonyl, carboxyl, amino, and phosphate. R groups represent aminos. You have oxygen, OH. So like take a look at phosphate, for example. It's charged. It ionizes to release the H, since phosphate groups can release H plus ions into the solution. This is considered acidic. An amino, however, is considered basic because the amino groups can actually remove the H plus, which is considered acidic, from the solution. So they're basic. Carbonyls are polar, methyls are nonpolar, hydroxyls polar, and if you want to just pause and get this down, this is going to be important to remember going forward. Take a second to talk about monomers and polymers. So macromolecules, which are what we're going to dive into in a second, they're large molecules made up of smaller repeating units. So you see this is known as a monomer, and then a polymer is the long chain made up of monomers. Monomers are the small subunits, and chains of monomers together make polymers, which end up forming the macromolecules. So what's important to remember with this is just that monomers make polymers, which make macromolecules. And then how they end up forming, we have hydrolysis and condensation reactions, which are the 2 different main types of how these molecules get formed. Condensation is the chemical process which 2 molecules are joined together to make a larger, more complex molecule with the loss of water. So it's important to remember condensation, loss of water. What it does is it squeezes the water out, which allows for bonding and allows for macromolecules to be created. Hydrolysis is the opposite, however, to condensation. A large molecule is split into smaller sections by breaking a bond, adding the H to one section and OH to the other. So going back to the functional groups, that's important. And hydrolysis is, you can break down the word hydro, water, lysis, splitting. So it ends up splitting a large molecule.
So carbohydrates are sugars. Most of the names end in os.
A macromolecule that we're going to go into is carbohydrates, sugars. So most of their names end in os, for example, glucose, fructose. They're classified by their number of carbons. The carbon is kind of the backbone to a sugar molecule. So their function is energy and energy storage. It's more of a short-term energy. Lipids are for long-term energy storage, but carbohydrates are good for short-term. Sugars are really good for short-term bursts of energy and energy storage in the body. Raw materials and structure, they're a sugar structure, usually 5-carbon, 6-carbon sugars. They form rings in aqueous solutions, and then from those rings you have attached hydrogens, and that ends up making the macromolecule. Their storage, they're stored as glycogen in animals, and they're stored as starches in plants. And as far as building, they build up as chitin in animals and cellulose in plants. And lipids, you can take a look at this diagram here. They have long tails. Lipids are your fats and oils. Like I was saying before, lipids are good for your long-term energy storage. They're comprised of your carbon, hydrogen, and oxygen. They're long hydrocarbon chains. They do not form polymers. They're in a fat structure, and the glycerol is the 3-carbon alcohol plus fatty acid, and the fatty acid with the long hydrocarbon tail with the carboxyl group head. So you can see that in this diagram. If you want to take a pause to take a look at this, that's going to be important moving forward. Proteins have, the thing about proteins is their shape really determines their function, and proteins have one of the most unique shapes of any of the macromolecules. You can take a look here. They form almost string-like kind of, they're all over the place, but their structure determining function is probably the most important thing to take out of biochemistry. And so just to take in the shape, you have your R group, carboxyl, and basically they're an organic compound, and they consist of one or more chain of amino acids. They form a peptide bond, which is a bond between amino group and one of the amino acid and the carboxyl group of another. As you saw, if you wanted to go back to the functional groups and go back to that diagram that I showed of the protein shape. And so shape determines function, and that couldn't be more true with proteins. The different shapes of proteins are what ends up giving them different functions in the body, which end up synthesizing different proteins for use throughout the body. And then polypeptide is a chain of amino acids, which is linked by the peptide bonds. So the peptide bonds is what you need to remember to associate with proteins. There's nucleotides and nucleic acids. So if you want to take a look, there's the nitrogenous base. So the nucleotides are in 3 parts: the nitrogen base, a pentose sugar, and a phosphate group. You can see that here as well as up here. And some examples of nucleic acids are RNA, which forms in a single helix, and DNA, which forms in a double helix. Nucleotides are the monomers of nucleic acids in DNA and RNA. So the nucleotides are made up of these 3 things: nitrogenous base, pentose sugar, phosphate group, and then these nucleotides are what ends up making nucleic acids, which make your DNA, RNA, which is the heritable information that the nucleic sequence encodes for. So that's that. And then so purines and pyrimidines are important to remember. Purine. So this is a pyrimidine, this is a purine, nitrogenous bases that make up the 2 kinds of the nucleotide bases in DNA and RNA. Types of these nucleotides are purines, which are a double carbon ring base. That's your A and G, that's your adenine and guanine. And then pyrimidines are your single carbon ring bases, uracil in RNA, thymine, U, TC, U, T. This is in RNA, and this is the TC is in DNA. So DNA replication, just a basic level of replication, transcription, translation on a biochemical level. Helicase is an enzyme. We're going to get into enzymes in a little bit. Enzymes that separates the double-stranded DNA into single strands to be copied by breaking the oxygen bonds between bases. That's very important because it allows other enzymes to go in and be able to help copy. Single-stranded binding proteins hold the DNA strands apart during replication. So during replication, the DNA unzips, held apart by the single-stranded binding proteins, and then DNA polymerase can come in and synthesize the DNA molecule from its nucleotides, as going back to the nucleotides and what they are. If you need to go back in the video to help understand that, now's the time to do it. And then primase is the enzyme that attaches to each strand and assembles a foundation for replication to begin. It's known as a primer. And DNA polymerase wraps around the single strand and attaches new nucleotides to the nitrogenous base. So just here you can see this is known as a leading strand, and this is known as a lagging strand, the Okazaki fragments. And so yeah, you can see just this DNA. This is important to copy down into your notes to help understand. So if you want to just take a minute to do that. And so enzymes. So what enzymes are biological catalysts that speed up chemical reactions. The substrate is, substrate and product are 2 big vocab words to understand with enzymes. The substrate is the substance which an enzyme works on, and the product is the substances which are produced by the reaction. One example of an enzyme is amylase, which is found in saliva. It breaks down the starch molecules into smaller glucose and maltose molecules. So it's just helping speed up the reaction that would already be occurring in your saliva. They don't cause the reactions, but they speed them up. Another example of an enzyme is lipase, and it breaks down fats into smaller molecules. Without lipase, it would take much longer to break down fats in the body. So enzymes are really important, and it's just important to understand what they are and what they do. And then, so metabolism and energy. When one of those chemical reactions takes place, energy is either taken or it's released. Now, your two vocab words for this are exergonic and endergonic. In an exergonic reaction, the energy is released to the surroundings. The bonds being formed are stronger than the bonds being broken. So that's important to remember and associate with exergonic. In an endergonic reaction, energy is absorbed from the surroundings. The bonds being formed are weaker than the bonds being broken. So then, taking that into consideration, the two types of metabolic reactions that take place in the cell, building up is anabolism and breaking down is catabolism. Anabolic reactions use up energy, so they are endergonic because they take in energy in order to use in an antibiotic reaction. Small molecules join to make larger ones. Catabolic reactions are what give out energy. They are exergonic, which means the bonds being broken here. And in a catabolic reaction, large molecules are broken down into smaller ones, which releases energy for the cells and body to use. So that's biochemistry in a brief overview. If you need to go back in this video at all to copy some of the diagrams or take your own notes, that would be definitely something I would suggest. Hope you enjoyed.